such as proportion.
Okay, so I'm handing out the review sheet for the final exam, which is on Wednesday of next week, 1 to 3 o'clock, and it's in this room. So this is the last week of lecture. We will not have lecture next Monday or Wednesday. So this review sheet just has a list of all the things that we have talked about, as well as essay questions that will appear on exam. Remember that the final exam is cumulative, so you also want to look at the review sheet for the midterm exam. But the essay questions from the midterm exam will not be on here. They'll just be these essay questions. Here's it. Okay, so I will have your last quiz graded and your homework assignment graded on Wednesday. So you'll be able to pick those up so you can study them for your final exam. Also remember that your formal lab report needs to be uploaded to get full credit tonight by midnight. After that, then I take 20% off for each day. Yeah.
For the extra credit, there's a submit assignment on campus as well. Was it one?
Extra credit? Oh, yes.
Okay, because like I saw it was due on Sunday, and I just made it in person.
No. Yeah. So your extra credit will also be uploaded to the Canvas shell. So there's a place to submit it as well. Are there any questions about that? Report? No. Okay. So we were talking about the structure of the ear. And so remember that sound waves have to travel through the ear canal or the auditory canal, and then they have to vibrate the tympanium. And that vibration of the tympanium is then amplified by the presence of the internal ear bones, which are called the malleus, incus, and the stapes. You'll also notice that from the stapes, information gets sent into the inner ear. And so I wanted to talk about the oval window as well as the round window. So the oval window is where sound waves enter the cochlea. Sound waves also have to exit the cochlea, and they exit via what is called the round window. So I'll just put in parentheses, they exit via the round window. Both of these are on the diagram that is in your textbook. Now the cochlea is actually filled with fluid that is very similar to your cerebral spinal fluid. And the sound waves have to pass through that fluid, and then they have to stimulate what type of receptors. So what type of receptors are involved in hearing? Does anybody remember? Are they thermoreceptors, mechanoreceptors, chemical receptors, chemoreceptors, mechanoreceptors? So the sound waves, remember, have to stimulate the hair cells. So remember, the hair cells kind of look like this, and they're all connected. So multiple hair cells are all connected via the cochlear nerve. So this is just showing that there's more than one hair cell that sends information out via the cochlear nerve. And then the cochlear nerve then transmits that information into the cerebral cortex. And so it goes to the auditory cortex. And the auditory cortex is where we consciously perceive the sound. So I could draw multiple hair cells here. Okay. So when we look at sound, there are different characteristics of the sound. So we could talk about the frequency of the sound waves. So the sound wave frequency is the number of sound waves in a given period of time. So if I was to just look at time on this axis, this would be a high frequency. This would be a low frequency. Okay. So this is a high-pitched sound. This is a low-pitched sound. The difference between pitches has to do with the location of the hair cells in the cochlea. So this is due to, this is high frequency, which is a high pitch. This is a low frequency. And so notice that there's only two waves here. So this is a low pitch. Okay. So if we look at the presence of those hair cells in the cochlea, there's a diagram in your book that shows this. And so the hair cells are kind of located right in the middle, and the sound wave vibrates this membrane that covers the hair cells. And then that vibration of the membrane causes the hairs to bend. And that causes transduction because the physical mechanical energy is turned into an action potential and then is transmitted out of the ear via the cochlear nerve. Now, if you look at the cochlea itself, different areas of the cochlea respond to different frequencies of sound. So if you look at the apex, which is the very front or very top of that, so if we look at the apex, that's 200 hertz. That would be a low frequency. And then near the beginning of the cochlea, we have like 4,000 hertz, and so that would be a high frequency sound. So where it vibrates the hair cells determines the pitch of the sound. We also have amplitude. So amplitude is the size of the sound wave. So if I look at a frequency, this would be one frequency. And if this is the same frequency, but the sound waves are shorter, this would be a loud sound, and this is a soft sound. So those have the same frequency. I tried to draw them the same frequency. So the same sound wave per given unit of time, but the difference is in the amplitude.
So is there a hertz that we can't hear low at, right?
Yes. And so some animals can hear much lower. Dogs can hear much higher, like the dog whistle, we can't hear it. Elephants can hear really low sounds that we can't hear. And so it could be when you're looking at an animal, they could be responding to sound waves that you are unable to perceive. Yep. Yep. And the sometimes with the amplitude, this can actually damage the hair cells. So I'll put can damage hair cells. And so the cells kind of get permanently deformed, and that is permanent hearing loss. So that's why it's important to wear hearing protection. Like if you're a carpenter and you're using loud tools all the time, that's why they have to wear hearing protection, or else they stop losing the ability to hear. Yeah.
Would it be possible that we could potentially be making sound that we can't even hear?
Well, if we're consciously making it, like speaking, I don't think that we are unconsciously making it, but obviously we're producing stimuli to other animals. So like maybe an animal can sense the sounds of our heart, sounds of our blood flow, sound, they might also be able to detect our electrical energy. But I think if I'm not consciously making sounds that I can't hear. So I could be unconsciously making sounds that I cannot hear. So if I'm a shark, like if I'm in the water and I'm going like this, you know, flailing around, they say that the sharks can sense your electrical currents in your muscles. So that would be something. So what were you thinking?
I was just thinking, like, I'm sitting in animal, but you didn't realize how you were somehow transmitting sound.
Oh, yeah. So unconsciously, yeah. Okay.
I have a question.
Yep.
So a frequency that you can't hear can have an amplitude loud enough that can damage your hearing even though you can't hear it.
No, because these hair cells will only bend at a specific frequency. So you don't have hair cells that bend at a really high frequency that you can't hear. So the way that you can tell if your hearing has been damaged is you probably all experience this. That's that fullness in the ears. Like you walk out of a club where the band is playing way too loud, right? And it sounds like this. It goes like that. Does everybody relate to that? Okay. So that means that you probably have done some damage to your ears. And I actually find the hand blowers in the bathrooms painful. Right? Their sound is way too high for me. So I'm like, when anybody hits the hairdryer, I'm like, Right?
Yeah, I can't listen to low bass noises anymore because it causes that fullness in my ears and my ears start hurting like that.
Yep. So if we look at that, this is what the damage to the hair cell looks like. And so this is permanent. And so this is why when you get older, sometimes you have to have a hearing aid that picks up the sound and then makes it louder so that you can hear it, right? Because of hearing loss. Okay, so we're gonna watch this really cool movie that talks about this. This is one of my favorite videos because it's just a really cool movie. It has a lot of information that you do not need to know, but it's really interesting, a lot of anatomical information they talk about. But it's really interesting because they actually demonstrate the vibrations. It's only like 6 minutes.
The sense of hearing is accomplished by a process known as auditory transduction.
Oh, and that's too low for me.
Convert sound waves in the air into electrical impulses.
Okay, let me see if I can get it up higher. Sorry about that. Having problems with my...
As sound enters the ear, it passes through the external auditory canal, where it meets the tympanic membrane. The tympanic membrane then vibrates in response to the sound. Sounds of a lower pitch or frequency produce a slower rate of vibration. And sounds of lower volume or amplitude produce a less dramatic vibration. Higher frequency sounds produce faster vibrations. The tympanic membrane is cone-shaped and articulates with a chain of 3 bones called the auditory ossicles. They consist of the malleus, the incus, and the stapes. The movements of the tympanic membrane vibrate the ossicles, passing on the information of frequency and amplitude. The 3 bones pivot together on an axis shown here in red. The pivotal axis is due to a series of ligaments which hold the bones in place within the middle ear cavity. The anterior malleal ligament and the posterior incutal ligament are of particular importance for the pivotal axis. 2 structures which normally obscure this view of the middle ear have been removed. They are the chordae tympani nerve and the tendon of the tensor tympani muscle. Through the ossicles, the vibrations of the tympanic membrane are transferred to the footplate of the stapes. The stapes moves with a piston-like action, which sends vibrations into a structure called the bony labyrinth. The labyrinth is filled with a fluid called perilymph. If it were a completely closed and inflexible system, the movement of the stapes would be unable to displace the perilymph, and therefore unable to send vibrations into the bony structure. Due to the flexibility of a membrane called the round window, the stapes movement can displace the perilymph, allowing vibrations to enter the labyrinth. The corridor leading to the round window is found within the spiral portion of the bony labyrinth, known as the cochlea. Vibrations produced by the stapes are drawn into the spiral system and return to meet the round window. The portion of the spiral passage in which vibrations ascend to the apex of the cochlea is called the scala vestibuli. The descending portion of the passage is called the scala tympani. A third structure, called the cochlear duct, is situated between the scala vestibuli and the scala tympani. The cochlear duct is filled with a fluid called endolymph, and when viewed in cross-section, the membranes separating the two fluid-filled systems are visible. They are Reissner's membrane and the basilar membrane. The membranes are flexible and move in response to the vibrations traveling up the scala vestibuli. The movements of the membranes then send vibrations back down to the scala tympani. A specialized structure called the organ of Corti is situated on the basilar membrane. As the basilar membrane vibrates, the organ of Corti is stimulated, which sends nerve impulses to the brain via the cochlear nerve. The actual nerve impulses are generated by specialized cells within the organ of Corti called hair cells. The hair cells are closely covered by a structure called the tectorial membrane. As the basilar membrane vibrates, the tiny clusters of hairs are bent against the tectorial membrane, triggering the hair cells to fire. The entire basilar membrane does not vibrate simultaneously. Instead, specific areas along the basilar membrane move variably in response to different frequencies of sound. Lower frequencies vibrate the basilar membrane closer to the apex of the cochlea, whereas higher frequencies produce vibrations closer to the base. This arrangement is known as tonotopic organization. Together, this sequence of events is responsible for our acoustic perception of the world around us.
Okay, so that is actually the answer to one of your essay questions. So if you look at your essay questions on your review sheets, you need a copy of the review sheets. Did you get wet? Did you get wet? No. Got it. Okay. So looking at question number 9, how are you able to consciously tell the difference between the smell of a flower and the sound of a ringing bell? Describe the stimulus. So in this case, it would be sound waves for the ringing of the bell. The receptors are the hair cells. The transduction is the bending of the hair cells causes an action potential to be transmitted via the cochlear nerve to the auditory cortex, where it is consciously perceived. Okay. So if you compare that or contrast that with smell, remember with the smell, we were talking about chemoreceptors in the nasal cavity bind odors, and the binding of the odor causes an action potential to be transmitted into the olfactory bulb, which then goes to the olfactory nerve, to the olfactory, actually it's olfactory tract, to the olfactory cortex. So the difference is where the action potentials are being sent and what kind of stimuli they are able to perceive, they're able to respond to. So remember, the transduction is just going from a stimulus to an action potential. And transduction occurs differently depending upon what type of receptor you have. Does that kind of make sense to everybody? So I mentioned previously that we also have organs of equilibrium associated with the cochlea. So hearing and equilibrium go together. And so you need to know the semicircular canals. You don't need to know the difference in what they do, but they oriented in the planes of our movement. So like this way or spinning this way. So they are also filled with fluid, so they can tell angular rotation, so what angle you're rotating at. There is also a part of the of this which is called the vestibule, and you don't need to know this, but I think it's kind of interesting. This is where you actually have what are called ear stones. So the ear stones are located in the vestibule, and they're called otoliths. And this actually allows you to determine your orientation to gravity and also vertical acceleration. So this is actually what allows you to sense if you're moving up or down in an elevator. So it also kind of allows you to tell whether or not you're accelerating forward in a car, right? Or when you stop suddenly, it gives you information about that as well. So the cool thing is that these are little ear stones called otoliths that then press down onto the hair cells that then will change direction when the force of gravity changes. Okay. So this is actually super important. And if you don't have this sensation, or if you have problems with your equilibrium, it can make you feel really bad. So imagine if you felt dizzy all the time. If you had vertigo all the time, people, like, it's so bad that people will sometimes, you know, end their lives, commit suicide, because that sensation of not knowing where you are relative to gravity or feeling dizzy and out of equilibrium is a really bad sensation. So these are the ear stones. They're different from the ear bones, right, which are the malleus to incus and the stapes. So sometimes trauma to the head can damage this and give people permanent vertigo. So it's a sense that is oftentimes underappreciated how important it is. Okay, so we are moving on to a new section, and we're going to be talking about skeletal muscle. And in lab, you got to see a slide of skeletal muscle as it is being innervated by the motor neurons. So we're going to look at how the motor neurons would stimulate a skeletal muscle, the skeletal muscle to contract. Okay, so you'll notice that skeletal muscle was striated. It tends to be multinucleated, which means that more than one stem cell fuse together to produce a muscle cell. And they are also voluntary. So unlike smooth muscle or cardiac muscle we talked about, which are involuntary, skeletal muscle is voluntary, which means that its movement is coordinated by the motor cortex. So we talked about the motor cortex versus the somatosensory cortex. Okay, So it is also, we tend to have these muscle cells. So a muscle cell is called a muscle fiber. And they are long because they're from the fusion of more than one cell together. So when you hear about, oh, fast twitch muscle fibers versus slow twitch muscle fibers, they're actually talking about this. This is a muscle cell. So within the muscle fiber, we have myofilaments. And the myofilaments are referred to as actin. These are proteins. Actin and myosin. So the myofilaments and their arrangements cause the skeletal muscle to be striated, and it also allows, it's these filaments that allow muscle contraction. So myo means muscle. Filament in this case means a protein. So this is why muscle tissue is so high in protein, is it's filled with myofilaments. Okay. The actin is considered to be the thin filament. And it has myosin binding sites. This is the thick filament. And these have what are called myosin heads that can bind to the actin filament. Okay. These myofilaments are arranged in what are called sarcomeres. So they are arranged in a sarcomere. Sarcomere. So if we look at this arrangement, I'm just going to draw one sarcomere, but there are hundreds of sarcomeres down the length of one long muscle fiber. So the sarcomere looks like this. We have the thin filaments connected to the central protein, that center line. And then we have thick filaments in the middle. Okay. Actually, I'm going to draw a third one. Sorry, a third one down here. Usually drawn in threes. Okay, so this is one sarcomere. So this would be the actin. This is my myosin. You don't need to know this, but this is called the Z disc. But this whole thing is called one sarcomere. So if I was to look at my myosin, they are what have the myosin heads. And so they kind of look like this. So these are these little structures that can actually pivot. So you would label these the myosin heads. And they can bind to the actin. And the way that a muscle shortens, and this was discovered in the 1960s, I have to say that before this whole model was discovered, which is called the sliding filament model, they actually thought that the proteins themselves shortened. But now they realize that the reason why the muscle shortens isn't because the proteins shorten, it's because they slide in relationship to one another. And so this sarcomere, when it shortens, it connects with these heads and it pivots and it causes the whole sarcomere to come in. So the sarcomere can shorten. So this was proposed in the 1960s, and the people that discovered it got a Nobel Prize for it. And it is called the sliding filament model for muscle contraction. So we're going to come back to this in a moment. But we're just going to put this into perspective as to where are these sarcomeres in relationship to like my bicep muscle. Okay. So in your book, there is a diagram that shows the relationship. So this is a bit of my biceps muscle. And if I was to open up the biceps muscle, it is actually an organ, because the muscle itself is encased in connective tissue, and then it is subdivided into fascicles, which are also full of connective tissue. So when you eat a steak, you can kind of sense that it is chewy, and it's chewy because of the collagen fibers and the connective tissue that are present. So you notice that this is a bundle of muscle fibers. This would be one muscle fiber right there. Notice that it is multinucleated. Notice that the nuclei are actually pushed out to the side because the cell itself is filled with these myofilaments. So this is my myofilament, right here. And this would be my sarcomere. And so this is a diagram that shows the size of the sarcomere. And notice that there are sarcomeres all along the length of the muscle fiber. And then the sliding of the filaments together is what causes the muscle to shorten. And we have, in some cases, the ability to do fine motor control. So I could like move my finger just a little bit, and only a few myofibers are contracting, or I can move my whole finger at once, right? And that's a lot of muscle fibers are contracting. So some muscles you can do this with, other muscles you can't. Like if you think about trying to just move your big back muscles just a little bit with fine motor coordination, it's just not, you're not capable of doing it, okay? So again, this is these just shows the myofibrils, right? The actin and the myosin. And it also shows the sarcomere. Okay. So let's look at more about this sliding filament model. So with the sliding filament model, the myosin heads bind to the actin. The heads pivot. The filaments slide towards each other, and the sarcomere shortens. So this pivoting of the heads takes ATP. So ATP is necessary. So that's our energy is necessary specifically for the unbinding of the myosin heads. And so if ATP runs out, the muscle will stay contracted, okay? So no ATP, muscle stays contracted. So what is that called in terms of the TV shows that you watch, might watch online? What is that called when a person has died and their muscles are contracted? Rigor mortis. Okay, so this is actually the basis for rigor mortis. So that means contraction after death, because ATP runs out and then the myosin heads cannot unbind, and so they stay connected and the muscles stay contracted. Now, rigor mortis could last a while, but oftentimes if decomposition actually causes it to go away. So it ceases decomposition, right? So decomposition would be where the bacteria and the fungi get into the body and start to break it down and cells start to fall apart. And so decomposition happens and rigor mortis ceases. Okay, so let's see if I have my picture here. Okay, so this is a diagram showing it in a little bit more complicated route. So notice, let's just start at the bottom here. So this is also in your book. So at the bottom, ATP has to bind in order for the head to release, but it also energizes the head. So when ATP, so let's talk about the role of ATP. ATP can be converted to ADP, which is just adenine diphosphate, plus phosphate, right, plus energy. So when ATP is broken down, it actually energizes the myosin head. So ATP energizes the myosin head. And ultimately powers the pivot, right? So if the head is energized, then when it attaches to the actin, it can use that chemical energy and convert it to mechanical energy, and it can cause the pivoting to occur and the filaments to slide. So showing a sarcomere that is okay, so this would be a sarcomere that is stretched out, and this would be a sarcomere that is completely contracted. This actually shows something interesting in terms of, like if you're interested in going into sports physiology. So one of the interesting things about stretching is it actually separates the actin and the myosin. So you could actually overstretch the muscle, and overstretching is actually going to decrease the power of the contraction. So if you are overstretched, when you go to lift a weight, you're not going to have as much strength because there's not going to be as many overlaps between the myosin heads here and the actin filaments. So imagine if this was even stretched further, that top one. Okay? And then if you're not stretched out at all, then your filaments could be all the way like this. And so then you can't produce much force either. So there is actually an optimal stretch. There's an optimal length of your muscle that will give you the most power when you contract. Right? So that's kind of interesting from a sports physiology, that you want to stretch but not overstretch if you're like interested in lifting the most weight. Yeah.
Does it have to do with like concentric, eccentric contractions?
No, well, actually it does, because like you can get really sore if your muscle is stretched and you're having to use it. You can get really sore. So, for example, if I'm walking up a hill, my quadricep muscle is actually stretched because my calf muscle is actually stretched, and then I'm having to power the walk. So I'm having to power the walk with a stretched calf muscle, and that will make your legs really sore. So if you're walking uphill, because you actually have to use more muscle in order to do the same amount of work. Yes, that's a good point. And that's eccentric muscle. Yeah. Okay. So now we're going to talk about how we regulate when a muscle will contract and when a muscle will not. Okay. So this goes back to the nervous system. So remember that we have a motor cortex, and this is the conscious control of our skeletal muscle. cortex, we have axons that extend out and down, and those are called motor neurons. So motor neurons innervate skeletal muscle. They can be both excitatory or inhibitory, right? So sometimes a motor neuron needs to relax one muscle so the other muscle can contract. And when did we talk about that? Or where you have to relax one muscle so the other one can contract. Anybody remember when we talked about that? That was in the knee-jerk reflex, right? So if I'm going to contract this muscle, I need to relax the antagonistic muscle so that they're not contracting in opposite forces, causing my leg to do nothing, right? So we have a prime mover, and the antagonistic muscle would have to do opposite of what your prime mover muscle is doing. So if I want to bend my arm, my nervous system has to say, triceps relax, biceps contract, right? So we also have to inhibit contraction. Okay. So these motor neurons release the neurotransmitters. So the neurotransmitters diffuse across the synapse. So the synapse is just the space between the motor neuron and the skeletal muscle fiber. They bind to receptors. If excitatory, it causes the depolarization of the membrane. So excitatory causes depolarization. So this depolarization is the same thing that we were talking about when we were talking about the depolarization of a neuron, right? So the excitatory causes the depolarization. Okay. This depolarization leads to the release of calcium ions in the skeletal muscle fiber. So they're actually stored in the muscle cell itself, and only when they're being stimulated do the calcium ions be released. Okay, so to be released by skeletal fiber, muscle fiber. So calcium is a necessary mineral. It's a necessary nutrient for muscle contraction. So if you don't have enough calcium, your muscles are not going to be able to contract. Okay. So these calcium ions, so right there, they're a positively charged ion, binds to a protein called tropomyosin. So tropo means upon. This is upon myosin, but it's actually the myosin binding sites of the actin. So the tropomyosin is going to the tropomyosin. So I'll put which blocks the binding sites on the actin. The binding of calcium causes tropomyosin to move. So this causes tropomyosin, and I think I forgot one thing. So this actually binds to a protein called, I have to go back up here, called troponin. Okay, So the calcium actually binds to troponin. Tropomyosin is what blocks. This will make sense when I show you a video in a second. So the tropomyosin changes shape and moves. To expose binding sites. Okay, so let me show you a picture of this in a second. Okay. So this is my motor neuron. These are my vesicles that are filled with neurotransmitters. Once an action potential gets to the end of my motor neuron, it can release that neurotransmitter. And in this case, they're showing acetylcholine is the neurotransmitter. So notice that the acetylcholine binds to receptors that are on the outside of the skeletal muscle. It then causes the depolarization, then causes calcium to be released. Okay. And then this causes, notice here, the troponin, which is this yellow shaped molecule, is not covering the sites in which the actin, or excuse me, the myosin head wants to bind to the actin. Here it is. So if acetylcholine is not released, then no contraction will occur because the myosin cannot bind to the actin. If calcium is present, it allows the binding sites to be present so that binding can occur and the filaments can slide. Okay, so the calcium binds to troponin, and this allows for the formation of what they call a cross bridge. Okay, so we're going to watch just a little animation video of this, which is only 3-4 minutes in length.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1, cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2, the power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3, cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4, reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
Okay, any questions about that idea? That was revolutionary, the idea of that sliding filament model. Explains a lot about the physiology of the skeletal muscle and what we know of how it works. You know, that's not something that we can see with our eyes. So a lot of this stuff is not something we can see with our eyes. It's just done through experimentation. You can see it with an electron microscope. You can see the sarcomere shortening. We can't see it with our eyes, but you can actually see the sarcomeres with an electron microscope. So that has a higher resolution than light microscopes that we use. Yep. But obviously, we can't generally see the, I don't think we can see the actin and, or no, the troponin and the tropomyosin. We can't see them. Yeah. Yeah. Okay. So we are going to now move, sorry, we're jumping around a lot into the quarter. We're going to move to reproduction. So I have a handout for you today with male and female reproductive structures. So you'll remember that we've talked about reproduction a little bit as it relates to other animals. So we talked about the difference between asexual reproduction, where you're producing clones of a single individual, and then sexual reproduction, where you're producing offspring that are genetically different than one another. Is there extra one? Yeah. So in sponges, for example, if they are going to produce the gemmules to prevent from dying during dehydration, that would be asexual. But remember that when we have the production of the egg and the sperm to produce genetically distinct offspring, that would be sexual reproduction. So reproduction can be asexual and sexual. And there's been a lot of discussion as to why most animals reproduce sexually and not asexually. And they believe it has to do with the fact that this increases the genetic diversity and allows us to co-evolve specifically with our microorganisms. So it allows for co-evolution with microbes. So asexual is essentially cloning. I'll put that because we generally think of cloning as being biotechnology, but animals themselves can just make clones of themselves. So we looked at one organism that was capable of asexual reproduction. And so what is this? What type of organism is this? Sea anemone, and it is in the phylum Cnidaria. And here it is in the process of budding, right? So it's just actually just breaking into two. And that would be an example of asexual reproduction. So remember that when you look at those sea anemones on the Oregon coast, oftentimes they are formed in colonies of genetically identical individuals. Okay. What about this organism right here? What phylum do you think it's in? Mollusca, because they're not really flat. They're sea slugs, and so they are in the class Gastropoda or Gastropoda, and so they are also called nudibranchs, right? So in this particular instance, these would be examples of hermaphroditic organisms that are producing both egg and sperm. And so in this particular instance, they both have copulatory structures, and this copulatory structure is just transferring the sperm. So if you think about the cost of sexual reproduction, one would be that you have to get close to another individual. There could be a cost if that individual wants to eat you, like when we talk about spiders, right? There could also be a cost because when you get really close to another individual, you could get disease from that individual, sexually transmitted diseases. So that's a cost. And the other cost is that sometimes you have to expend a lot of energy to reproduce. So for example, you have to compete for access to mates. You might also have to dance. You might also have to produce bright plumage. You might also have to use a lot of energy to coerce the individual to reproduce with you, right? So in some insects, the males provide a nutrient package. They actually provide food to the female before the female will let them reproduce with them. Okay. So there's lots of costs to sexual reproduction, so there obviously must be the big benefit, which is an increase in genetic diversity. Seems to be super important. Okay, so we also have mechanisms of sex determination. So this is what determines whether or not you are a male or a female. And in us, this is genetic and it is chromosomal. So this would be at one type of mechanism. So this is a genetic mechanism. Oops, I spelled that wrong. Chromosomal. I'll spell that again. So this is a genetic mechanism. So we have sex chromosomes and we can have two copies. So this would be the female, and in us, this is the male. So this is in mammals. Birds, interestingly, have a different mechanism. They actually have different sex chromosomes. So in birds, the one that differs, they have Z and W, and this is actually the female, and then the ZZ is actually the male. So this would be in birds. So notice that in this case, sometimes they say it is the father that determines the sex of the offspring, because the sperm either have an X chromosome or a Y chromosome that fertilizes the egg. And so the male determines the sex. In this case, it would be the female, because an egg could either have a Z or a W. All the sperm have Zs, right? So it's just a different, somehow it evolved differently. Okay. We can also have environmental mechanisms. Okay. So one thing that can affect sex determination in a lot of reptiles is temperature. So this would be like reptiles. Okay. So if the incubation temperature is high, so they incubate their leg, incubation temp high, like it would be in the middle of a nest of eggs, then they could become a female. And if the incubation temp is low, then they become males. And I can't remember where, but I read something about how they're worried about some species of reptiles because of climate change. They're all becoming either female or male, depending upon which temperature. So if it's high temperature, they're all born female, and then there's no males, and it's like that's going to be the end of the species because of their inability to respond quickly enough to these changes in temperature. So in some it's vice versa. So in some species, it's the opposite of that. And then sometimes it's just what the other individuals are doing. So what other individuals are. So in some species, we have what is called sequential hermaphrodism. So sequential means that they're male and then they become female. So this happens quite frequently in fish, right? So if you're a male and everybody else is a male, and you're like, wait, there's no females around, right? Maybe you should become a female, right? So they have the ability to change sex based upon what other individuals in the population are doing. So sex is more fluid than maybe you thought, right? It's sex determination. Okay, so we are going to focus on mammalian reproductive structures. So this is the description of the male reproductive structures. So when we look at the what is referred to as the gonad, gonad, these are the testes. The gonad is where the sperm are produced. So this is function would be sperm production. The testes also produce testosterone. So they're actually also an endocrine gland. So they're part glandular because they also produce testosterone. Now, the testes in mammals is not located up in the body cavity when the male is reproducing. So in some species like rodents, for example, the testes can be up in the body cavity when they're not reproducing, and then when they get ready to reproduce, the testes descend into what are called the scrotal sacs. Okay. Now, the function of the scrotal sacs, I would argue, is not protective. If you think about protecting the gonads, they should be in the body cavity, right? Not outside of the body cavity. So I wouldn't say it's very protective. What do you think the function of the scrotal sacs are? Yes, they are temperature regulators. So for some reason, sperm production needs to not be as warm as it is up in the body cavity. So it needs to be about -3 degrees Celsius. So that would be like 34 degrees Celsius if your body temperature is 37 degrees Celsius. So it needs to be cooler. If the temperature is too warm, the sperm are deformed. So the scrotal sacs have the ability to pull the testes up towards the body cavity to warm it up. And they have the ability to let them down, to cool them down. And so they always recommend if you're trying to, if you're like a couple and you're trying to get pregnant, that you allow the scrotal sacs to move, right? So that you wear baggy underwear, right? If you have really tight underwear on, that's going to keep the scrotal sacs warm, and they're not going to be able to regulate their temperature very well. And the sperm could become not productive if they're too warm. Okay. So that's the scrotal sacs. Now, surrounding the testes is a structure which is called the epididymis. This is actually where the sperm become mobile. So they have the ability to swim. And so this is also where the sperm are stored. So sperm become mobile. Oops. Become mobile. And they're stored here prior to ejaculation. Now, Now, from the epididymis on ejaculation, the sperm have to be transported into the body cavity. So the sperm have to go into the body cavity, and they travel via a structure called the vas deferens. This is also sometimes called the ductus deferens. So this transports the sperm into the body cavity. Has to go into the body cavity because in males it has to hook up with the urethra, and then it has to come out of the penis. It also has to pick up nutrients and important other components of the semen. So let's look at the diagram that I handed out to everybody. Okay, so this would be the scrotal sac. This is the testis, which is singular for testes. This right here is my epididymis. So that structure that surrounds, kind of looks like a C surrounding. And then this tubule right here is the vas deferens. Now, it's really confusing because the vas deferens does not travel alone into the body cavity. It actually travels in the same group of its muscles. It's also nerves come down, blood vessels come down into the testes. And so this travels via the spermatic cord. So travels via spermatic cord. So one way that you could decrease the fertility, right, is to cut the vas deferens. So when they talk about a vasectomy, right, ectomy means to cut. This is that you're cutting the vas deferens. So this is cutting the vas deferens, so that the ejaculate should not have sperm in it, but it will still you'll still have an ejaculate when you have a vasectomy because of all the other things that are going to be added to the duct before you get to that point, okay, before it comes out the urethra. Okay, so that would be a form of sterilization, right? So that you couldn't reproduce. Sometimes it doesn't work though. Sometimes it automatically kind of comes back together. So it doesn't have 100% effective rate, but it's pretty close. Okay, so let's look at the glands. So we have what are called accessory glands. So the first accessory gland is a paired gland called the seminal vesicles. And I always want to put two S's in there, but I'm pretty sure there's only one. Vesicles. Okay. So these actually produce about 70% of the semen is from the seminal vesicles. And the semen, the seminal vesicles add fructose, which I think is a very strange thing. I don't know why they don't just add glucose. Fructose is a slightly different, it's an isomer of glucose. It's generally just found in fruit, hence its name, fructose. But this is another place where it's found, and this is what the sperm use for energy. So this is the energy source for the sperm. So the sperm need to have access to fructose so they can produce ATP and so they can swim. It also produces a coagulant. And this actually thickens the semen upon entry into the vagina. And the reason for this is that this prevents backflow. So the once ejaculated into the female's tract, you don't want it to just flow back out. In some species of rodents, the coagulant is so effective that it actually produces what is called a reproductive plug. So the female actually gets plugged up by the semen. So if another male tried to ejaculate into the female's reproductive tract, it would not go in because she's got that plug. But the problem is that eventually the sperm need to swim. And so we'll talk about the next gland, which is called the prostate gland. There's only one of these. It's not paired. So the prostate gland produces, one of the things that it produces is an anticoagulant. So this thins the semen so that they can swim. So it has its action after ejaculation into the female's reproductive tract has happened, right? So the semen or the plug will start to thin a little bit so the sperm can swim. The other thing that the prostate gland produces, which is interesting, are prostaglandins. Now, this is a really common chemical that is actually produced by a lot of different tissues. But in this particular instance, it actually serves to cause smooth muscle contractions. So it causes smooth muscle contractions in the female's reproductive tract. So in the female. So you want the vagina and the uterus to kind of contract to help move the sperm into the reproductive tract. So that those contractions actually aid in the movement. Okay, and maybe that's why it's called the prostate gland, because it produces prostaglandins. Now, if we look at the location of these on my diagram, this would be my seminal vesicle. This is one of them. This is the other one. Vesicle. This is my prostate gland. For some reason, people in the United States, men in the United States, have a lot of prostate issues. This is not the case for other places. So it could be our diet, it could be our lifestyle. There's something wrong with the way that we age. Prostate gland, you'll notice, is near a structure called the urethra. So this is my urethra right here. And the urethra drains my bladder. So I'll put the bladders up here. So you'll notice that the urethra has to travel through the prostate gland. So almost every male, when they get to be 70 or 80 in this country, will have prostate problems. And so it might make it very difficult for you to urinate. So older men have to constantly go to the bathroom because they can only urinate a little bit at a time. And it can also make that urination very painful. It could also lead to problems with ejaculation because you'll notice that the vas deferens comes around, comes past the seminal vesicles, picks up the stuff, and then it also goes through the prostate gland. So one thing that they sometimes do for males is that they go in and they actually just increase, when the prostate gets swollen, they'll actually just go in and increase the diameter of that duct by kind of coming in and clearing it out so that males can urinate. Prostate cancer is also a big problem, but they actually believe that you're more likely to die. If you're an older man and you get prostate like near your 70s, you're actually more likely to die of something else than your prostate cancer. And so oftentimes they just don't even treat the prostate cancer because they figure you're going to die before it becomes an issue and spreads. So there's one more gland here that we didn't talk about, and that's this structure right here. And this is called the bulbourethral gland. So the bulbourethral gland produces pre-seminal fluid. Okay. So in general, it does not have a lot of sperm, unless an ejaculate has happened recently and there's still sperm in the urethra. So the function of the pre-seminal fluid is to neutralize the acidity in the urethra. So it neutralizes acidity in urethra, and it functions to lubricate the penis. The acidity could be different in the urethra because remember, sometimes we get rid of extra hydrogen ions by secreting hydrogen ions into our filtrate. So sometimes our urine could be really acidic, and that could kill the sperm. The sperm die when the acidity gets too high. And so that just kind of neutralizes and gets rid of any urine that might be left in the urethra. So this right here, this would represent the blue parts here, represent the erectile tissue. So remember that we talked about the parasympathetic nervous system actually causes erection by vasodilating, causing dilation of those blood vessels so that blood will flow into that tissue and will actually cause the penis to become stiff. Okay. Females also have erectile tissue. It's in the clitoris, and then the clitoris is actually a little bit larger. It's actually has its biggest part is on the inside where you can't see it. Okay. So the erectile tissue. In most mammals, I have to say, in most mammals, the penis actually has a bone, and it's called a baculum. And so, like cats and dogs and even whales, they have a bone that is in the penis, which helps with its keeping it erect. Okay. So that's the male reproductive. Okay. So we'll just start with the female, and then we'll finish up with female reproduction on Wednesday. Okay. So the gonad. Oops, gonad. I can't spell gonad today. It's funny. Gonad are the ovaries, right? This is where the egg is produced. Then we have what is called the oviduct. Sometimes these are also called fallopian tubes. Now, the way that our reproductive system develops is that really early on, like within the first 5 weeks, you can either become male or female. So you actually have an oviduct and a vas deferens, and then one deteriorates while the other develops when you decide which sex you're going to be. And so the oviduct is unique in that it does not directly connect to the ovary. So when the egg is released from the ovary, there's just a brief moment where it's actually in the body cavity. And then the oviduct has to suck it up. It has to suck it into the tubules. And this is actually where fertilization takes place. So in the oviduct, this is where fertilization takes place. Then the uterus is where the implantation of the embryo occurs. If it occurs in the fallopian tube, that is bad. Sometimes it does, and it's called a tubal pregnancy. And they actually have to go in generally and remove that. And it usually damages that side of the reproductive tract, so they actually just kind of tie off the fallopian tubes on that side. And it could actually, if it ruptures, it could actually lead to death in the female. So the implantation of the embryo should occur in the uterus. And then separating the uterus from the vagina is called the cervix. So this is a barrier between the uterus and the vagina. And then the vagina is what receives the male's copulatory structure. So this receives the penis. And that would be where ejaculation occurs. Okay. So let's just label those parts on the female reproductive tract. Okay. So this would be my ovary. Notice that the tubule has like these finger-like extensions, and those are actually called fimbriae. And the egg gets ovulated, and I'm going to show you a video on Wednesday about this. So it ovulates, and then there's cilia lining the fallopian tubes that suck the egg into the oviduct. So this is my oviduct. This would be my uterus. Notice how muscular it is. That's all smooth muscle. This is the barrier, so this is my cervix. And then this would be the vagina. The other thing I'll just point out on here, this is the vestibular gland. So it produces secretions that lubricate the vagina. Notice that it is homologous, it's actually in the same location as the bulbourethral gland is in males. Okay. And then this right here would be the erectile tissue. And I'll show you a better picture of this, erectile tissue. And then this exposed part is called the clitoris. I'm just going to label in here, this is the urethra. So remember that in females, the urethra is completely separate than the reproductive tract. So the urinary tract and the reproductive and the feces, the digestive tract are three different openings, where in the male, the reproductive and the urinary system have fused, right? So that it just comes out in one opening. Okay, so I will stop there for today. And then we will talk more about female reproduction. We're also going to talk about reproductive cycles on Wednesday.
Yes.